morning. I'm really excited to introduce Dr. Anastasia Lambrou, I hope I pronounced that correctly, and Dr. Phil Shirk. Dr. Lambrou is an Epidemic Intelligence Service Officer in CDC's Respiratory Virus Branch. She's an infectious disease epidemiologist with a background in biosurveillance, pandemic preparedness, and global health security. She obtained a Master of Science in Epidemiology from the London School of Hygiene and Tropical Medicine and a Doctor of Philosophy in Global Disease Epidemiology and Control from the Johns Hopkins Bloomberg School of Public Health. Dr. Philip Shirk is a Presidential Management Fellow at the CDC, and he works on optimizing influenza viral surveillance for improving flu vaccines. And for COVID-19, he's been working to produce data that feeds the CDC's COVID variant tracker website. That I assume it's that dashboard that I presented the first thing that I showed this morning. And he has a Master of Science in Biology from the Virginia Commonwealth University. And a Doctor of Philosophy and Zoology from the University of Florida. So we're really excited to have them here today. So please take it away.
Thanks so much for having us. Everyone can see my screen. And yeah, it's perfect that you started on the BA.2 estimates because yes, that is what we do every week. And we're going to kind of delve into those methods. So thanks for that introduction. We're going to be presenting on the SARS-CoV-2 genomic surveillance program. So how does data get in to make those estimates? And then Philip will tackle how we actually create those estimates. Perfect. So I'm going to get started on the overview of the system and then pass it to Philip to introduce our variant estimation methods. And then he'll take a deeper dive into those methods, looking at early Omicron emergence and how we ended up estimating that. So before jumping into the details of the genomic surveillance program at CDC, why conduct genomic surveillance? So I've included some of the examples of how we use the data. So first, for situational awareness at the state, regional, and national level. What variants are circulating and at what levels? The genomic surveillance data also informs the strategic allocation of medical countermeasures like monoclonal antibodies. The surveillance data also supports effectiveness studies, especially when new variants are emerging. Genomic surveillance can also elucidate introductions of new variants and transmission dynamics. And we've also seen a lot of use at the local level for contact tracing and other epidemiologic investigations. So let's start with a general overview of some of the major parts of the genomic surveillance program. So first, patients are sampled, and these come into the surveillance program from a variety of sources, testing programs, commercial pharmacies, public health labs, and they are sequenced by different partners, most external to CDC, and that data is then brought together to be modeled and the output presented for public health action at different levels. So CDC's genomic surveillance program is comprised of three major sources of sequence data that feed into those estimates. So first is the National SARS-CoV-2 Strain Surveillance Program, also called NS3, in which CDC has partnerships with state and local public health labs that submit random samples to CDC for sequencing. The second source is contract sequencing data in which CDC has partnered with commercial diagnostic laboratories for sequencing. And lastly, this third source is comprised of tagged metadata. And these are sequences uploaded to public genomic data repositories like GISA, GenBank, by state and local public health labs, universities, or any non-federally contracted diagnostic lab. So these three sources come together to comprise the National SARS-CoV-2 Genomic Surveillance Program. At the start of the program back in November 2020, only the top first pathway of NS3 existed, in which samples were sent to CDC for sequencing from state and public health labs. These samples are also integrated into a viral repository at CDC for further downstream study. Then as the Alpha variant emerged in January 2021, the second pathway comprised of contract sequencing was stood up. And then most recently, in October 2021, we stood up this third pathway, which incorporated public health labs, academic, clinical, and other labs that were not part of the top two pathways, in which they can tag their sequences in genomic repositories for use in our surveillance program. So starting on the left here, after sampling, these samples are sequenced. They go through initial bioinformatics along the three pathways I just described. And all of these consensus sequences come together, are merged, deduplicated, filtered by geography, checked for quality, and then enter our bioinformatics and modeling pipeline. And then variant proportion estimations, which Philip will present on, are published each week on our public COVID data tracker website. So we produce these variant estimations weekly, and we move through this cycle. So throughout the week, our sequencing partners are submitting sequences, and those are being ingested into our system. Everything that arrives before Thursday, we'll pull in and analyze that data to be included in that week's analysis. Thursday is analysis day, and Friday we check in on the models and estimates. And then each Tuesday, you'll see on the CDC COVID data tracker website, these estimates are updated. At the start of this year, we took a look back at the genomic surveillance program and how it's grown, and we've even created new pathways. So between June 2021 and January 2022, over 1.1 million sequences were submitted to the surveillance program, which was an average of approximately 35,000 sequences per week. And looking to the previous time we had evaluated the growth in the system, it had grown 3 times since that previous evaluation that looked at December through May 2021. Thinking back to those 3 pathways I had shown about, where does the data enter the surveillance system, approximately 2% of data comes from NS3, 88% comes from contract commercial labs, and 10% comes from that tagged sequences. The surveillance program has also received sequences from 56 jurisdictions, and that includes all 50 states, DC, American Samoa, Guam, Northern Mariana Islands, Puerto Rico, and the US Virgin Islands. So I'm going to pass the baton over to Philip, who's now going to present our variant estimation methods and some specifics on modeling the emergence of the Omicron variant.
Good morning. Here on the left is a plot showing how the number of sequences available for analysis has changed over time. And on the right is a map showing the geographic variation in where these sequences originated. Next slide. So one of the ways that we deal with this variation, temporal and geographic, in the number of sequences that are available is that we use a complex survey design with a weighting strategy to help account for that. Another challenge that we face in estimating variant proportions is that the steps Anastasia went over take time. It usually takes 2 to 3 weeks before most of these sequences go from being collected as a sample to ending up in our database. So for the most recent 2 weeks, the current week and 1 week into the past, we use a multinomial regression model to nowcast estimates of variant proportions. And a third challenge is that we are not directly measuring variant proportions. We are modeling and estimating variant proportions. So there is uncertainty in those estimates. And we present 95% confidence intervals and 95% prediction intervals for the multinomial model to help display that uncertainty in our estimates. Next slide. Here's some more information on the survey design that we use. Our primary sampling unit is the source laboratory that did the sequencing for each sample. We stratify by state and week, and then we weight samples such that each sample's weight represents the number of infections that are represented by that individual sequence. And this survey design is adequate for estimating variant proportions once we have enough data, so 2 or more weeks into the past. Next slide. When we have less data, so the most recent 2 weeks, the current week, and 1 week into the past, we use a multinomial logistic regression model to estimate the variant proportions. And we use 2 different models that are very similar, one for estimating the national variant proportions. And for that model, we have variant, categorical variable, as our response variable, and then week, a continuous predictor variable. Our second model is a regional model that has the same response variable, the same week predictor variable, plus a categorical region predictor variable. And these are the HHS regions that you see in maps, or that I will show you soon if you haven't seen them already. Next slide. So here is what these estimates look like on the COVID data tracker website. It sounds like you already went over this a little bit this morning. I missed that as I was trying to log into Zoom. But on the left, we have a bar plot of the different variant proportions. And if you click on one of these bars, so the rightmost bar is currently selected as indicated by the arrow, the triangle underneath the bar. Then on the right-hand side, you can see the point estimates for each of these variants, as well as the 95% prediction interval. And this is showing the national estimates, but you can also get these same figures for different regions. Or if we go to the next slide here, you can see maps of the different regions, HHS regions, so that you can visually compare the proportions between regions across the US. So this is what we do on a regular basis. I'm gonna switch topics here and talk a little bit about our estimates from December, when our Nowcast estimates were not as accurate as we would have liked. So if you go ahead to the next slide, here we go. This is showing the number of sequences that were available in different weeks in December and early January. And as you can see, as we've already mentioned, the current week, which is week 0 on the x-axis, has very few sequences available that were collected in that week. 1 week into the past, there are some sequences available, but well under 10,000 sequences in each of these individual weeks. Whereas 2 weeks into the past, there's well over 20,000 sequences available for each of these weeks. And so that is when we start using the complex survey design alone to estimate the variant proportions. Here, I'm going to be calling these estimates weighted estimates, which can be a little bit confusing because we use the same weights and the same survey design in the multinomial Nowcast model. But just to contrast it to the Nowcast model, I'm going to be calling these weighted estimates. So if we go to the next slide here, you can see what our weighted estimates looked like throughout the month of December. And in different colors here, we have the estimates that were made in different weeks. And you can see that they're extremely similar. The estimates don't change much from week to week. Once you get above 20,000 sequences, adding in another 5,000, 10,000 sequences really doesn't change the estimate very much. On our next slide here, we're going to see what that looks like for the Nowcast estimates over the same time period. So the Nowcast estimates are shown in different colors here. And in the background, there's a dotted black line with the gray uncertainty bars that is showing the weighted variant proportion that was made in mid-January. So once we got more data in, that was our best estimate of what the Omicron proportion actually was at that time period. Whereas the Nowcast estimates were the estimates that we made before that data came in. And I'll point out a couple things here. First of all is that all of these models agreed that Omicron was starting out December at a very low proportion and was going to be the overwhelming majority of cases in the US by the end of December. And in that regard, all of these models were accurate. However, on a more quantitative note, a number of these estimates were less accurate than we would have liked. In yellow here is the estimate that was made the week ending December 18th, and that overestimated the proportion of Omicron in the United States. Once more data came in and we realized that it was an overestimate, we could then look at why our Nowcast model was misleading. And The reason that the Nowcast model overestimated Omicron in that week is because several of the labs submitting sequences to the CDC were reporting Omicron sequences faster than they were submitting non-Omicron sequences. And so the Nowcast model was projecting that forward and overestimating the growth rate and predominance of Omicron. However, 1 week later, so the line in green here, our estimate for the week ending December 25th, the Nowcast model underestimated the true proportion of Omicron. And what happened there was exactly the opposite. Most of the labs submitting sequences to us were submitting Omicron sequences more slowly than they were submitting non-Omicron sequences. So the first few sequences that came in those first 2 weeks made it look like Omicron was much lower. As more data came in 2-3 weeks later, we then saw that the proportion of Omicron was actually higher than our Nowcast model had estimated. Next slide. So this emphasizes the importance of data quality. Our multinomial model is assuming that the data are randomly sampled and representative, and it is extrapolating based off of the data that we give it. So it's very important to make sure that our data is randomly sampled, that labs are not prioritizing different variants, either to sequence them or to submit them faster. And another important component of data quality that we won't really get into here, but is also a big concern, is genome completeness, particularly coverage of the spike protein, which is a big indicator of how our immune system is going to respond to that virus. And so some sequencing approaches are more resilient and more consistent across a wider variety of variants than some other sequencing approaches. And finally, we need to continue emphasizing the uncertainty in our estimates. And that uncertainty is indicated in the confidence and prediction intervals. And it's also indicated by some data quality flags that are outlined by the National Center for Health Statistics. So an example here is that if your estimate is based off of a very small effective sample size, then that estimate receives a flag indicating that it's a relatively low quality estimate. Next slide. So in conclusion, sequencing takes time, and that delay is a large determinant in how we are able to estimate variant proportions. When we have a large amount of data, we can use the weighted survey design to estimate variant proportions, and those estimates are quite stable. When we have less data, we can use the Nowcast estimates to project the variant proportions, but those estimates are pretty sensitive to bias in reporting speed. And unfortunately, we can really only determine whether or not there is bias in reporting speed retrospectively. When we're actually receiving the sequences, it's really hard to know if those sequences are representative of what will be submitted in the future or if they will not be. And so with that, I'm going to hand it back over to Anastasia for a couple more slides.
So we could go on about genomic surveillance all day, but we wanted to close on some resources for those of you that might be interested in our methods or in genomic surveillance further. So first off, Philip and I led an MMWR that came out in February that has the updates to the program that we described and also compares the emergences of the Delta and Omicron variants. This report looked at genomic surveillance from January 2021 to January 2022 to get that year-long picture of the dynamics. Second, we have updated our GitHub with detailed methods, model equations, our code of the methods that we presented today, and also an example data set to see our methods. And lastly, the CDC Genomic Epidemiology Toolkit is a great resource for anyone who wants to apply genomic data to public health action, from examining clusters to wastewater and early warning. And lastly, it really does take a village to keep a surveillance platform running, models running, and we have an amazing team of people here to acknowledge. So thanks for having us, and yeah, looking forward to your questions.
Thank you so much for that really nice presentation and for all the, I assume, immense work that has gone into all of this. I know these aren't easy undertakings. I was wondering if you could give us a little bit of information about how maybe weekly the data are being used by the CDC public health officials or distributed to kind of state public health officials, and what policy decisions are potentially being kind of decided or made on kind of like a weekly or bi-weekly basis.
That's a great question. I think we spoke a lot about what goes to the public. So internally, these are sent around as well. They also feed into the governor profile report. I'm not sure if you've seen that report, or the community profile report that's also online, which has case data, hospitalizations, social vulnerability index data, and then also genomic surveillance, and hopefully soon wastewater as well, to kind of get that holistic picture of what does the pandemic look like now in this week. Also, a lot of interagency sharing of results. They come to meetings that we have on Fridays, and also that allocation of medical countermeasures, especially monoclonal antibodies, as we're learning about effectiveness in real time. A lot of communications with BARDA and ASPR on how to do that as well. I don't know, Philip, if you want to add anything else from your side of things.
I think that's good. I think you know more about this topic than I do, so I'll defer to you on that one.
Thanks. Other questions from the group?
Thank you both so much. And I apologize, I came in a little late if you mentioned this, but I'm curious what the relationship is between sort of your operation at CDC and the kind of data that are coming in to your analyses and decisions that are being made on a state or even local scale about which specimens to sequence, how many to sequence, from what geographic areas. Like, how does what your needs sort of trickle down to those like very local decisions?
So also every Friday we have something called TOAST. We love acronyms, in which it's basically technical office hours where CDC and then state and local public health labs meet every week to discuss what should we be sequencing, is anyone having issues with their protocols, what are our focuses right now, what are needs at our level, their level, to keep that communication open, because sometimes it does differ. And if a state is looking at a particular outbreak, CDC might only want a random sample or one of those samples from that outbreak, where the state would like to further investigate and do phylogenomics within that cluster. And so what to send to CDC, then what's helpful for local public health action. And I think the biggest question we get at those Fridays calls are, how do we sample and what types of sampling schemes are helpful? So I think you hit the nail on the head there about when we need to conduct random sampling, when would it be useful to get all sequences from a particular cluster, depending on what the public health question is, because it does vary even at regional levels as well. And I think this is also another reason, I'm not sure if you've seen this, but CDC has launched the Pathogen Genomic Centers of Excellence that will pair public health labs and state public health with an academic institution or another sequencing institution to build capacity at the local level and also answer important questions using genomic surveillance data. So that call just came out 2 weeks ago.
Great. And I actually have a couple more questions, and of course, jump in. Yeah, thank you for that. So is there much, we have learned so much about respiratory viral data, surveillance, forecasting through COVID that, has some obvious implications for how we should handle flu going forward. Is there any crosstalk between, or maybe you guys are the ones doing both, but is there much crosstalk within the CDC about how to handle molecular surveillance for flu in light of what we're doing for COVID? Are similar tools, your weekly discussions, are they covering both viruses? How does that relate? How do the two responses to both viruses interacting in-house?
That's a great question. I was a flu epi person in a previous life, and now Philip's coming into flu at CDC. So we talk a lot about flu. And we do see a lot of the response is focused on SARS-CoV-2, and teams are response specific and virus specific. But now that things have been opening up a bit more, the transition in conversation is to pan-respiratory. And so those conversations are happening, thinking about surveillance, not just genomic surveillance, thinking about syndromic, hospital-based, community surveillance, going more pan-respiratory. It would be great one day to even see pathogen agnostic, right? Looking flexibly at public health threats and pathogens. But the conversation is happening now about broadening things to multiple respiratory pathogens, and not even just flu and SARS-CoV-2, but the whole spectrum.
Great. Okay, interesting. Okay, my last question is, I mean, you guys are eat, sleep, and breathing this genomic surveillance data, and probably think about it even more than we do as we think about COVID night and day. What is your personal take on what's happening with BA2 in the United States, and whether we're going to see an uptick, a meaningful uptick in any or all of US states?
I think we get that question a lot, even for like the last 3 weeks or so. I don't know, Philip, if you want to start on this. I know everyone has their own personal hypotheses as well, so I'd love to hear from the room. But, you know, I was surprised that we didn't have a larger uptick right now, even just given S gene target failure data, looking at genomic data, and some counties that have even 80 to 90% BA.2, their wastewater seems stable, cases seem stable, but I do think we're going to start to see upticks. And it'll be a big mix of, I think, some of the factors that even Spencer brought up earlier, both host factors and previous immunity and that initial Omicron wave and how much that hit the total population, as well as what we're doing now, just practice-wise. I know that's, you know, a little bit of not a standard answer, but I don't know, Philip, do you have any ideas as well?
I've got to admit, I don't have many opinions on this either. I think that's kind of the big question. Why do so many countries vary so drastically in terms of how quickly BA2 grew and took over? And in the US, it's slowly gained proportionally, but it's gained proportionally as the case counts have dropped precipitously. So at this point, it's not really causing much uptick in case counts. It looks like it might in the near future, but I think we're just going to have to wait and see.
Yeah, it is interesting to just keep looking at Europe and where they are in this, you know, new mini wave. And it's also kind of a new case study globally, seeing what's happening in China, right, or Shanghai, or where immunity might be different and what BA.2 is doing.
Yeah, thank you both for your personal takes on it. I really appreciate it. Looks like Darlene, you have your hand up. Go for it.
Yeah, thanks so much for your presentation. You mentioned wastewater sampling, and I was wondering whether it's gone beyond just sort of indicating presence or absence in certain communities, and whether you're able to get at quantitative measures and even able to sequence what you find, and how well that might track with what you're seeing in your own surveillance with the sequencing.
So one caveat is that Philip and I are on the clinical side of the sequencing team. There is also a wastewater team. However, we also meet with them and collaborate. I know the majority of their sites right now are more PCR amplicon based and aren't sequencing as much, but they have a plan to incorporate long-term sequencing into that system to get more at genetic changes. And I know that they are actively working on quantitative metrics and also linking that to other epimetrics like hospitalizations and other parts of the surveillance system and integrating that together. So sorry, I don't have details on that, but I know a lot of updates are coming out on that side.
Yeah, no, that's great. And if I understand correctly, some of the mutation panels that they're running might give you a sense of the variants found. And so I wonder if you guys have compared, you know, from week to week, what those measures sort of look like, or what those predictions look like on your side with what they're finding in the wastewater.
No, definitely. And trying to get at what that early warning window is. I think all different teams across the globe are converging on a time window, but yeah, trying to cross compare has been really helpful, especially with BA.2.
And I will note that another early warning indicator is PCR tests with S gene target failure can indicate in the past the difference between most Delta variants and most Omicron variants, and currently can pretty reliably distinguish between BA2 and other Omicron variants. So there's another kind of early warning indicator there that can use the results of PCR tests, and we don't need to wait for full sequencing data to be available.
Right.
That's a great point, Philip. And we had talked before about public health decision making. And once again, all of this data coming together for decision makers. S-gene target failure has been one of the other sources as well.
Right. Do you guys see challenges as more people move to at-home testing with your surveillance program and sort of the ability to keep your eyes on what's going on and have this early warning system in place?
That is definitely a concern. Exactly how it's going to influence our estimates is unclear at this point. We are reviewing, currently the testing data does play a role in the weights that we use for weighting our sequences for estimates. And so as the testing data changes, that could influence the weights. Some comparisons of a variety of weighting methods suggest that that likely won't strongly influence the estimates, but we are currently reviewing other options for moving forward once the testing data does change more drastically because of that testing data.
Thanks so much.
So I don't know if it's just been luck or maybe something else, but I kind of have a question that, so it seems like the kind of dominant variants that have been detected don't seem to have emerged in the United States. And I guess my first question is like, do you have any hypotheses for why that is, given, we have such high infection counts that, that there seems like enough room for that to happen. And then my follow-up question is, in that kind of bioinformatics process you described, how would it work if there is some, like, is there a flagging process for like, this is, a variant that is concerning, I guess, based on like the sequence sample obtained, or does that happen like more upstream, like you mentioned, with like state investigations of specific outbreaks?
So I don't pay a ton of attention to these. I do know that other people are looking at the mutations, the protein mutations or protein alterations that occur in the spike protein, to look for potential variants that would be more concerning based on how similar they are to current vaccines and other variants that are in circulation. So that is definitely a consideration. And there was a second part of your question that I just forgot.
I think it was the US side of things, right? I'm going to piggyback off of what Philip just said real quick. The flagging process is kind of throughout the process. So the first big flag is sequence quality, but also if a pango lineage isn't called, going back to look at potentially why. And so we're not part of that team, but there is a team that looks specifically at mutations, like Philip said, and will flag in that early part before it goes further downstream. But then if you want to repeat your other part of the question, that'd be great.
Sure. I'm actually going to ask a different question, which is, so in that process, it sounded like states were coordinating the outbreak investigations, but I remember for some of the influenza surveillance, CDC was actually helping to direct, like, we noticed this, like, send us more samples or things like that. Is that also happening, or is that kind of not centralized at the CDC for COVID?
No, yeah, CDC tries to play a technical assistance role as much or as little as a state requests. So on day-to-day work, now where we are at in the pandemic, states will just submit their sequences. If they have questions, they let us know. We will help try and confirm, like with Omicron, and some states didn't have their bioinformatic pipeline up yet to confirm that with high sensitivity. We would help confirm that. Sometimes we go through periods of enhanced surveillance. which I think used to happen with flu potentially, where we would look for if you have breakthrough cases or this potential mutation and then work more directly with states as compared to the week-to-week normal submission process.
Thank you. So Darlene asks if you are supporting sequencing programs in other countries as well, like technical support, I assume, as well.
Yes, we definitely are. And someone that Philip works closely with that built this weighting scheme was actually supporting the Afro region during Omicron as well about weighting and sampling structures. And I think there was also a bioinformatics training a couple of weeks in February. So lots of work going on the technical side of sequencing and building capacity to the bioinformatics side to the public health action. This is what it says for BA.2 or for Omicron. You know, what do we do next? How do we interpret?
So one kind of maybe final question I have is, you know, the vaccines, I think initially for COVID-19 were discussed in a way that there was like plans to quickly update them as new variants of concern emerged. And I was wondering if there's, you know, a process being put in place at the CDC. Philip, I think you work a bit on like influenza vaccine kind of strain selection ideas. Maybe I misinterpreted that from your bio, but so I was wondering is, you know, do you see COVID-19 moving in the direction of flu, where there will be kind of these maybe annual summits to develop what vaccine or what strains should go into a vaccine booster? Are those happening already? And if not, do you think it's time for those to happen, or, you know, just continue along with the current vaccines? I'd love any of your thoughts related to
that. Yes, so on the influenza side of things, again, I'm working on similar things in terms of aggregating data and estimating proportions and summarizing the data that we have, the surveillance data that we have, so that we can make more informed decisions about what goes into the annual influenza vaccine. On the coronavirus side of things, I think the conventional wisdom, the prevailing opinion at this point is that coronavirus will become endemic and will require pretty regular booster shots unless kind of community immunity kind of builds up to where people have had enough infections or vaccines in previous years that you have some natural immunity, as people call it, to future infections. But I think a lot of that is pretty up in the air right now. And I will say that I don't have the best answer for that question. This is my understanding of things, but Anastasia,
I think with the pandemic response, we've learned how important this interagency collaboration has been because we all have different roles in that vaccine part with like CDC and surveillance, or NIH and research and platforms, or BARDA, ASPR on the, you know, platform side of things. And now that we do have these platforms like, you know, mRNA vaccines, like monoclonal antibodies as well, which could be tailored genetically in real time for multiple pathogens. These systems are being stood up to open these, create, you know, communication panels between CDC and what's going on the genomic side, to what is going on in the research development side, to the neutralization studies that are going on, right, as an ecosystem. So yeah, I'm hoping to see that continue in the future, and for all respiratory pathogens, as we discussed before.
Thanks. Any final questions from the group? All right. Well, thank you both so much for joining us and for the wonderful presentation, and, you know, letting us ask a bunch of questions and great responses.